Hello everyone. This lesson is going to be on two-trait or dihybrid crosses. Unlike previous crosses that we've taken a look at, this one here is not only going to look at one single gene and one single trait at a time, but as the name implies, two different traits. And as we'll see, these two different traits will be for two different genes, and they will have to be on separate chromosomes as well. In fact, this does depend upon the genes being on separate chromosomes. And if you think back to the process of meiosis, we did see this process of independent assortment that does take place during metaphase 1 of meiosis, and this is where the chromosomes do line up independently of each other. So again, what we have taken a look at already are single trait crosses. Let's just say that we have the A gene and that an individual, a diploid parent individual, has two copies of the A alleles, and in this case, I'm just saying a capital A and a lowercase a. Keep in mind that these are just on chromosomes, so why don't we say that it's on chromosome pair number 1, and on one of the chromosomes, the paternal chromosome, we find the capital A, and we'll say that on the maternal chromosome, the one coming from mom, there is a lowercase a. So same chromosome number, homologous chromosomes, similar chromosomes, but not identical. They both have the same gene, the A gene, but one has the dominant version and the other one has the recessive version. So what we know already from single trait crosses, this would be a single gene for a single trait. And what we would then know is that in the gametes of course are not going to be diploid, they are going to be haploid, and they will have either one or the other chromosome, meaning they would have either the capital A or the lowercase a, but not both of them. But when we're talking about two traits, and these two traits, two different genes, are on separate chromosomes, we'll draw another one here, and we'll just say that on chromosome number 2 now, we'll make this a little bit longer, and we'll have on one of the chromosomes, again, I'll make it the paternal one that has the dominant one, and I'll just call it the B gene, and the other one, the maternal one, again, the same chromosome, homologous chromosomes here, the same gene, only different versions of it. This one will have the lowercase. So now this is still a diploid cell. There are two copies of each chromosome. There are two copies of each gene. And the way that we would write the genotype for this particular adult would then be capital A, lowercase a, keeping the A's together, writing the capital letter first, and capital B, lowercase b. But if we do have these genes that are now on separate chromosomes, again, they are going to assort independently of each other. So when we do have the gametes that are formed in this case, we can have the capital A that ends up in one gamete along with the capital B, and that would give us a haploid gamete, which has one of the capital A's and one of the capital B's, one of each of the letters, and again, that would be haploid rather than the diploid parents. The other possibility is the other two chromosomes would go in the opposite direction, and we would end up with another gamete with the lowercase a and the lowercase b. And again, these gametes are haploid because they have one of each letter. But of course, keep in mind that these genes, they are on separate chromosomes, and these chromosomes, they do assort independently of each other. So just because we do have the capital A that is going in one direction, that doesn't mean that we have to have the other paternal chromosome, the capital B, going in the opposite direction. So if we do have our capital A, and if we just say that it again is going over toward the left, it's just as likely that we could have the lowercase b that goes in the same direction. Now we would have a gamete that has, again, one of each, the A and the B's, but now they have the capital A and the lowercase b, whereas the previous one that I showed you here is the one where it had both of the capital letters going together. I'll just write that one again. We had the capital A and the capital B going together. And if we do have the capital A and lowercase b going in the same direction, what that means, of course, is that we would have to have the lowercase a now going with the capital B. We've been writing the A's first, so we'll continue to write the A's first. And this is now the fourth gamete that we have. So from this one genotype, two different genes for two different traits. We can get four different possible gametes when the diploid parent is heterozygous for both of them. If you remember this equation that we talked about before, 2 to the x, where x is the number of heterozygous genes, both of these genes are heterozygous. 2 to the 2, four different possibilities. These are the four different possible gametes. 1, 2, 3, and 4. Of course, there's an easier way to do this, an easier way that I did show you before, rather than drawing these chromosomes and showing the separation of the chromosomes. That again was really just to remind you of independent assortment. But if we did have a parent that had this particular genotype here, an easier way to figure out the different possible gametes is to simply say, well, we can get the big and the big together, big A and the big B. We can get the big A and the little b together. We can get the little a and the big B together, and then we end up with exactly what we had before, these four different possible gametes. Again, this one, as a reminder one more time, is a diploid genotype. And on the right-hand side, the ones that I've circled, those are the haploid genotypes. For any of these two trait crosses then that we are going to be talking about, the two different genes, the two different traits, it is critical. This only works, two trait crosses, if in fact we're talking about two separate chromosomes for the different traits and the different genes. So how we arrange these and how we make our predictions is once again using a Punnett Square. And we're just going to follow exactly the same rules that we did before. If you're not given the letters, you need to assign the letters that you're going to be using. Again, keep in mind that a capital letter is used for the dominant trait, lowercase letter for the recessive trait. Write down the parent genotypes. Those, of course, are going to be diploid. Determine the different gamete genotypes, which are going to be haploid. And from that, you're going to complete your Punnett square. You'll see that it can be a significantly larger Punnett square, up to 16, rather than the 4 Punnett or 4 squares that we've been dealing with so far. And finally, the ratios for these ones here, the ratios, it's usually only the phenotypic ratio that we are interested in. And we'll see that when we get into taking a look at a couple of crosses. So we will, in fact, start by taking a look at some of the crosses that Gregor Mendel would have done. So Gregor Mendel's garden peas, all the different traits that he did take a look at. For this first example here, what we're taking a look at are two different genes. One gene is for the flower position. The flower position can either be along the length of the stem, that is what is called axial, and that one is dominant. The other possibility is the flowers are toward the end. That is called terminal. If axial is dominant, then that means that terminal is recessive. Choose the first letter of the dominant trait. We use A for axial, and now we're going to use not a T, but a lowercase a, the same letter, only lowercase, for the terminal flower position. The second trait that we're taking a look at here, completely different trait and on a different chromosome, and that is for the height of the plant. So they can either be tall, which is dominant, or short. Capital T then for the dominant tall trait, and a lowercase t for the recessive short trait. So now we have assigned the letters that we are going to use. Now we can actually take a look at the cross that we're being asked to perform. So it says a plant known to be homozygous dominant for both traits. Two different traits, homozygous, the same letter, and dominant for both of them. So keep your A's together. So for the flower position, it would be homozygous axial, big, big. And for the height, homozygous dominant tall, big, big for that one as well. So that would be the genotype for the first parent that is homozygous dominant for both traits. The other parent, exactly the opposite, homozygous recessive for both traits as well. So now instead of big, big, it's little, little for the axial and also little, little for the flower position. So whereas the first one was axial tall, these plants are of course going to be terminal and short. So let's just fill that in again for the parents. So it was the big, 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 and all lowercase letters for the other parent. The gametes for these ones, relatively straightforward. From the first parent, all they have are capital A's, which means that every single one of the gametes will end up with one and only one A. They will not end up with two of them. They have to end up with one, and again, it is only one. They only have capital T's, so they will all have a capital T. This is the only possible gamete genotype that we can get from parent number one. From parent number two, similarly, we can only have one possibility for the A, and that's a lowercase, one possibility for the T, that's a lowercase t. This is the only possible gamete genotype that we would have coming from parent number two. So now for our Punnett square, this is a larger Punnett square than what we have seen before. And what we now need to put in each one of the columns and rows is once again the gamete genotype. But keep in mind, now we have it not just for one gene, but for two genes. So I'll put from parent number one along the side. So we can write for each one of these, big, all along the side. But in reality, of course, they're all the same, all of those. So for these ones, we can really neglect these final three rows here. From the other parent, homozygous recessive for both. So again, we could put that in each one of the columns, the top of each one of the columns. But again, we really don't need these rows here. It's just kind of repetitive. For this first cross, all we need is the one single square. So now we're going to fill in. This is our prediction for the next generation. What does it say? Well, from the first parent, they're going to get a capital A. From the second parent, they're going to get a lowercase a. From the first parent, a capital T. From the second parent, a lowercase t. Every single one of the offspring in the F1 generation are going to have this particular genotype heterozygous for both of the genes. And in terms of the phenotype, all of them are going to be axial and tall. So that is more or less our ratio. All of them heterozygous for both traits, all of them axial tall. That's not all that terribly difficult, but now what we're going to do, make it a little bit more challenging. This is the F1 generation. Now we're going to do the F2 generation. So these were the offspring. Now they are going to become the parents for the next generation, which means now what we're crossing are two parents that are heterozygous for both of the genes. And now we'll find that we do actually need the entire Punnett square. So where I have here is P2. Those are just the parents in the second generation, but those were the offspring from the first generation. Again, this is what we have. Heterozygous for parent number one, heterozygous for parent number two. So if you recall what I did on that first slide when we had the A's and the B's, I'm just going to do the same thing with this one, only instead of the A's and the B's, I have the A's and the T's. Each parent in this case, because both of them have the same genotype, are going to generate exactly the same combination of gamete genotypes. And that's going to be the big, the little big, and the little. Let me write that at the top here. Big A, big T is one possibility. Big A, little t is our second. Little a, big T is the third, and little is going to be our final possibility. So I am going to go through here and I'm going to fill in the entire Punnett square the way that I have written it at the top, starting with the big, ending with the little. I'm going to do that going across, and I'm going to do the same thing going down in exactly that same order, and you'll see the reason for that very shortly once I do get this entire thing filled out. So it takes a little bit of time. Be kind of careful. Try not to make any mistakes when you are filling in all of these letters. And it'll just take me a minute or so to do just that. So why am I writing the A's first and the T's second? Well, in the original question, it did talk about the flower position first, and that's why I'm writing it first. If it talked about the height first, then I would write the T's first. You need to keep the A's together. You need to keep the T's together. For each individual gene, the A's, it's the capital first, then the lowercase. And for the second one, once again, the T's, it's the capital letter first and then the lowercase. So again, the reason for me writing it in this particular order, starting with the big, big, little, big, little, and the same thing down the side, is now what I can do is I can circle all of these ones right here, except for this one. So all of those that I've circled, they have some things that are in common. They all have a capital A. Doesn't matter what the second one is, they're going to be axial. They all have a capital T. Doesn't matter what the second one is, they're all going to be tall. All of these are axial tall, and if we count them up, there are 9 that are axial and tall. Now, if we take a look at that one that I circled in the middle, and if I circle this one here and this one here, just to make it a little bit clear, I'll underline those ones as well. So all of these ones, they have something in common. Also, they all have at least one capital A. The second letter doesn't matter, but the T's are both lowercase. In other words, these ones are going to be axial short, and there are 3 of these. If we take a look at and circle these ones right here, all of those do have 2 lowercase a's. So they are going to be the ones that are terminal in terms of the flower position. And they're also going to have at least one capital T. Second letter doesn't matter. They're going to end up being tall. And there are 3 of these. The final one that we do have, tucked right in the corner here, the only one out of the 16, that is lowercase for both of them, and that is our 1. So this does give us, if we do rate it as a ratio, it gives us a 9 to 3 to 3 to 1, and this is the phenotypic ratio. Again, the genotypic ratio, don't bother writing all of those down. We'll just go by the Punnett square itself. Now, keep in mind that whenever you tell me a ratio, you need to tell me what that is for. So the 9 was the axial tall. One of the 3's was the axial short. The other one was the terminal tall, and the last one was the terminal short. Don't forget to tell me what each one of those numbers does represent in terms of the phenotype. This is a very important phenotypic ratio. This is the phenotypic ratio that you get when you are doing a 2-trait cross and both parents are heterozygous for both traits. So this is an important one, kind of similar to a previous one that we saw for a single trait cross. Single trait cross, we saw that there was a very important 3-to-1 ratio. This was for a single trait cross if both parents are heterozygous for that one single trait. And in this case, the 3 ended up being the dominant phenotype. The 1 ended up being the recessive phenotype. So that's an important one to remember, the 3-to-1 single trait cross, both parents are heterozygous. The 9-to-3 to 3-to-1 2-trait cross, both parents are heterozygous for both traits. Finally now, just a couple of other ratios that are relatively common, and if you do recognize these ratios and have an idea of what you should be looking for the genotype, it'll make things a little bit easier. So if it's a 1-to-1 to 1-to-1 ratio or a 3-to-3 to 1-to-1 ratio, in both of these cases, one of the parents, again, is heterozygous for both traits. If it's a 1-to-1 to 1-to-1 ratio, though, the second parent is homozygous recessive for both traits. So you can set this up, but what you will notice is that because you only have one possibility for one of the parent or one of the gamete genotypes coming from one of the parents, you only need 4 different Punnett squares. And because you only need 4, that's why you end up with this 1 to 1 to 1 to 1 ratio. The other one, the 3 to 3 to 1 to 1 ratio, again, one of the parents is heterozygous for both. The other parent is heterozygous for one of the traits, but homozygous recessive for the other. You do need to be kind of careful, though. The one that I've circled in red here, it is the B gene that is recessive, homozygous recessive, but there could be the other possibility where it is the A gene, which is homozygous recessive, and it is the B, which is the heterozygote form. So again, these are fairly common ratios that you will come across. The more common one is the 9 to 3 to 3 to 1 ratio, but it's kind of nice if you do recognize these other ones, the 1 to 1 to 1 to 1, and the 3 to 3 to 1 to 1.